Today we're going to examine a cellular model of neurodegeneration. We'll explore research that uses a protein called Progerin to simulate aging in a laboratory setting. The goal is to better understand its relationship with another protein, tau, which is central to certain brain diseases.
A key observation in the study of neurodegenerative disorders is their strong correlation with age. The risk for conditions such as Alzheimer's disease increases substantially as we get older. This suggests that the cellular environment of an aging brain may be a critical factor in the development of these diseases.
This brings us to a significant methodological challenge. How can a process that unfolds over a human lifetime be studied in a laboratory? Researchers require a model system that can replicate key aspects of cellular aging on a much shorter timescale.
So, let's begin by outlining the specific challenges researchers encounter when attempting to model age-related brain disease in vitro.
There are several primary difficulties. Natural aging is a slow and multifactorial process, making it difficult to pinpoint specific causal mechanisms. While useful, animal models do not always fully replicate human pathology, and perhaps most importantly, it's a challenge to isolate aging as an independent variable to directly study its contribution to disease.
To address these issues, the researchers in this study developed an approach to induce an age state in cells directly.
The tool they used is a protein known as Progerin. This is a mutated form of lamin A, a protein involved in maintaining the structural integrity of the cell's nucleus. In humans, this mutation is the cause of a rare genetic disorder characterized by accelerated aging. In the lab, however, Progerin can be used to induce a cellular state that mimics certain aspects of aging.
So what does Progerin do within a cell? It disrupts the nucleus, leading to instability and DNA damage, which are considered hallmarks of cellular senescence. A critical point for this study is that expressing Progerin alone does not cause the tau protein to aggregate. This allows researchers to study the effect of the aged environment on tau pathology as two separate variables.
This brings us to the core of the experiment, where the researchers combine their model of cellular aging with the introduction of pathogenic tau. They focused on a process called tau seeding. This is a prion-like mechanism where a misfolded tau protein acts as a template, or a seed. It causes healthy tau proteins to adopt the same misfolded shape, leading to the formation of aggregates that can spread from cell to cell. The experimental setup was as follows. First, they used cells engineered to produce a tau protein tagged with a yellow fluorescent marker. Next, they introduced the pathogenic tau seeds, which were extracted from mouse brains. Finally, using microscopy, they could automatically count the number of cells that developed fluorescent aggregates, giving them a quantitative measure of seeding efficiency.
Now we'll examine the outcome of combining the Progerin-induced aging with the introduction of tau seeds. The results were informative.
The data from this experiment was quite clear. As this chart shows, the cells expressing Progerin, the aged cells, displayed a significantly higher frequency of tau aggregation compared to the control cells. This effect was observed consistently in two different clones of the progeric cells, indicating that the aged cellular state enhanced the susceptibility to tau seeding.
This finding led the study's authors to conclude that premature aging and pathogenic tau can have an additive effect within their model system. One stressor appears to potentiate the effects of the other.
As with any scientific finding, it's necessary to consider and rule out alternative explanations for the observed results.
One possibility was that the progeric cells simply produced more of the fluorescent tau protein, which could lead to more aggregation. However, measurements showed that the amount of tau reporter protein was the same in both the aged and control cells.
Another alternative was that the aged cells were more permeable to the external tau seeds. To test this, the researchers measured the efficiency of introducing a different protein into the cells. They found no difference, which suggests the increased aggregation was due to an intracellular process, not a difference in uptake.
With the primary finding established, the next question was a mechanistic one. What was happening inside the aged cells to make them more vulnerable to tau seed?
To investigate this, the researchers employed proteomics. This technique uses mass spectrometry to measure the abundance of thousands of different proteins within the cell, providing a comprehensive snapshot of the cellular state under different conditions.
The proteomic data revealed a clear pattern. When exposed to either Progerin alone or tau aggregates alone, the cells showed some signs of stress but seemed to manage. However, when both stressors were present simultaneously, there was a significant disruption to the cell's protein quality control systems, accompanied by extensive deformation of the nucleus.
In their analysis, the researchers used a fold change threshold of greater than 1.5 times to identify proteins that were significantly altered, ensuring they focused on meaningful biological changes.
The pathways most affected by the combined stress were those related to protein quality control. This includes the endoplasmic reticulum stress response and the ubiquitin-proteasome system, which are responsible for identifying, tagging, and degrading misfolded proteins.
This evidence suggests a mechanism where the combined stress from cellular aging and tau aggregation overwhelms the cell's protein degradation machinery. When this system is compromised, the cell is less able to clear the pathogenic tau, which likely explains the increased aggregation observed in the aged cells.
Finally, let's consider the broader implications of these findings for our understanding of neurodegenerative disease.
The primary implication of this work is that the aging cellular environment is not a passive backdrop for disease. Instead, it appears to be an active factor that can amplify the underlying pathology associated with proteins like tau.
We can trace a plausible sequence of events. The process begins with the Progerin protein disrupting the integrity of the nucleus. This nuclear instability appears to compromise the cell's protein quality control systems. With these systems impaired, the cell is less equipped to handle pathogenic tau, leading to enhanced accumulation.
This research points toward a potential therapeutic avenue. If instability of the nuclear envelope contributes to this vulnerability, then strategies aimed at stabilizing the nucleus could potentially increase the brain's resilience to tauopathies during aging. It's a compelling hypothesis that warrants further investigation.